The life cycle of Leishmania in the human host. The life cycle begins with a female sandfly insect, which is infected with Leishmania, biting a human host. Following the ingestion of blood, the metacyclic promastogotes are released and enter the host through the bite site by regurgitation. Observe that the parasite reaches the mammalian skin and subsequently invades different cell types that it encounters, such as dendritic cells, fibroblasts, neutrophils that are rapidly recruited to the bite site, and mainly the macrophages. In this example, the attachment of the promastogote form of the parasite to the macrophage surface is observed. The process of internalization via phagocytosis begins with the formation of pseudopods. During and after the internalization of the parasitophorus vacuole assembles. In the vacuole, the promastogote transforms into an amastogote. Following the transformation, host cell lysosomes migrate and fuse with the parasitophorus vacuole. The lysosomal content is released in the vacuole. However, the parasite is not altered. Note that most amastogotes become attached to the membrane of the vacuole by their posterior portion, and the amastogote begins to divide multiple times. Observe how many amastogotes are accumulating due to the division process. The parasitophorus vacuole can occupy the entire cytoplasm of the host cell and culminates with their bursting out of the cell. The amastogote reached the extracellular space and subsequently appeared inside monocyte or free in the bloodstream. Thus, they may be sucked by another female sandfly during blood meal or invade new macrophages. Now observe in this example the attachment of the Leishmania amastogote form to a new macrophage surface. Note that the parasite is internalized via phagocytosis with the formation of pseudopods. During and after the internalization of the parasitophorus vacuole assembles. Observe that the host cell lysosomes migrate and fuse with the parasitophorus vacuole. The lysosomal content is released into the vacuole. However, the parasite is not altered. Note that most amastogotes become attached to the membrane of the vacuole by their posterior portion, and the amastogote begins to divide multiple times. Observe how many amastogotes are accumulating due to the division process. The parasitophorus vacuole can occupy the entire cytoplasm of the host cell and culminates with their bursting out of the cell, releasing the amastogotes. Thus, they may be sucked into a new female sandfly during blood meal or invade new macrophages.